We're starting a new chapter on viruses, and I added this chapter this year just because, well, we've had a global pandemic involving a virus. So I thought it timely that we at least learn a little bit about the structure, makeup of viruses, and how they reproduce. So what we're going to be looking at in this first little section here is just characteristics of viruses, kind of differentiating between what we call an enveloped and a non-enveloped virus, and then looking at the taxonomy of viruses, since they're not in our normal taxonomy of living things. They kind of have their own little structure. So first of all, the word virus is Latin for poison, and for a long time they couldn't see viruses just because we need an electron microscope to be able to view them, and so they knew something was just causing people to get ill and causing problems with their first research was with the tobacco mosaic virus, where it was causing disease on the tobacco plants. And so they came up with this word because it's like something was poisoning the plants. We consider them an obligatory intracellular parasite. That's because they have to have a living host in order to multiply. They can't multiply on their own. The other thing unique about viruses is that they have a single type of nucleic acid. They are either DNA viruses or RNA viruses. They can't be both at the same time. Their basic structure is that they have a protein coat that surrounds a nucleic acid. And some of them can have an outer envelope made of lipids, proteins, and carbohydrates, and we'll talk about that in a minute. They have to take over the host cell's machinery in order to reproduce, and then they help synthesize structures that help the virus transfer their nucleic acid to another cell. So they have ways of being able to get from one host cell to another. Now, viruses have a specific host range. That means that they only infect certain types of cells in only one host species. So it's not like a particular virus can infect a wide variety of different living things or even a wide variety of different cells in your body. Usually they're very specific to the host and to the type of cell in the host that they affect. We give the name of bacteriophage or a phage to viruses that infect bacteria. And so that word will be used quite a bit, so you need to get used to that. And again, they're very tiny, 20 to 1,000 nanometers in length, so you have to have an electron microscope in order to see them. When we look at viral structure, we use the term virion to basically be a mature virus. It's a complete, fully developed infectious viral particle. It's going to have that nucleic acid surrounded by a protein coat. Okay, and it's now ready. Once it's a virion, it's now ready to go from one host cell to another. All right. The nucleic acid, like I said, it has to be either DNA or RNA, but it can never be both at once. Okay, they can't have both like we can in our cells. The other unique thing is they can be single-stranded or double-stranded. So you could have single-stranded DNA or double-stranded DNA. You could have single-stranded RNA or double-stranded RNA. So that's a little bit different from our DNA and RNA because we have double-stranded DNA and single-stranded RNA. The capsid is the protein coat that surrounds the nucleic acid core, and it's composed of protein subunits called capsomeres. So it's all these little different individual capsomeres that make up the entire capsid or protein coat. And it's the arrangement of these capsomeres that's very unique to different types of viruses, and so that's one way we can identify them. Finally, we have what's called the envelope. Now, this is only in some viruses, and it surrounds the capsid. It's made up of proteins, lipids, and carbohydrates. And some animal viruses, when they're released from the host cell, they actually get coated with a layer of the host cell's plasma membrane, and that's what becomes the envelope for those animal viruses. The other thing is that the envelope can be covered in spikes. These spikes are made up of carbohydrates and proteins, and they project outward from the envelope, and so you can see all these spikes here, okay? If you've ever seen all the pictures, this is a picture of depicting coronavirus, but if you've ever seen any of the pictures of the coronavirus, they have all the different spikes sticking out from them. It looks like a round little ball, like a pin cushion or something with a bunch of pins projecting from it. These spikes help the virus attach to the surfaces of their host cell, and it's also a good way of identifying a virus and distinguishing one type from another. Non-enveloped viruses do not have an envelope. We call them naked viruses. So that's another term for them. Okay. So here's an example of a naked virus here. All you have, here's your capsid, and each of these individual things here are the capsomeres. Okay? And together they make up the capsid, that outer coat. And then you've got your nucleic acid. Could be DNA or RNA. Well, here's your capsid way inside here, that protein coat, but then we have this envelope surrounding it, and it can have these little spikes sticking out too from it. When we look at the shapes of viruses, the two main kinds that they can be, and it's based on what the shape of their capsid looks like. They can be helical viruses. They're kind of cylindrical in shape with a helical structure. So here you have this cylindrical shape. It's kind of hollow inside because that's where the DNA or RNA would be, inside there in that hollow part. Rabies, Ebola virus, those are examples of helical viruses. An enveloped helical virus is influenza, and so there's good examples of some of those. But then what many virus shapes are what we call polyhedral viruses. That just means they're many-sided. In fact, the main shape is what's called an icosahedron, meaning it has these 20 triangular faces and 12 corners. So you can see here's kind of one triangle there, another one there, and so on. It keeps forming, okay? So the adenovirus is an example of that. The herpes simplex virus, and there's various strains of that, anywhere from causing cold sores or warts to chickenpox and shingles, the different varieties. But this is an example of an enveloped polyhedral virus. And finally, we have the complex viruses where the bacteriophage would fall into there. And you notice that this looks like almost like a little astronaut's docking module or something here. They have these little tail structures, these little projections. These are what act as receptor sites that will connect to the receptor sites in the host cell. So they're almost like little docking stations. You have the head up here that's made up of the capsid and the nucleic acid in there, and then you have this sheath of it going through here in the tail part, and that's what will help inject the DNA through into the bacteria cell that it's going to infect. As far as taxonomy, you can look again on your book on pages 375 and 376 where it goes into it a little bit more detail. But they're grouped into family names or families based on the type of nucleic acid they have. Are they DNA or RNA viruses? How do they reproduce? And what is their shape? Okay. Now here's the thing you have to look at is the ending of the name. If it ends in virus, that's going to be the genus name. If it ends in viridae, that's the family name. And if it ends in ales, it's the order name. Okay, so you can identify it based on those endings of the name. When we use the term viral species, we're just talking about groups of viruses sharing the same genetic information and the same host range, meaning they would infect the same host and probably the same type of cell in the host. And usually they have more descriptive common names like human immunodeficiency virus, the virus that causes AIDS. Okay. So that's the first little part here for Chapter 13 on viruses.